Good morning, student. In last lecture, we have studied about mitosis. Now, we'll see another type of cell division, that is meiosis. Division of reproductive cell. We'll see that is meiosis. Meiosis was first demonstrated by Edward van Banden in 1887 and was described by Winnewater in 1990. Then the term meiosis was first coined by Farmer and Murray in 1905 from two Greek words meom to reduce and osis means state. Then this term meiosis was coined by J.B. Farmer and J.B. Murray in 1905. It is alternatively known as reduction division. Nuclear division takes place in two different phases, which are meiosis first and meiosis second. We'll see definition of meiosis. Meiosis is characteristic division of reproductive cell in which the chromosome duplicate only once but the cell divide twice. So the parent cell produce four daughter cell, each having half the number of chromosome and amount of DNA present in the parent cell. Hence meiosis also called as reduction division. It involves successive nuclear division occurring one after the other, while meiosis first and meiosis second. So meiosis first as reduction division, which result into two haploid cells, which form two haploid cells, and meiosis second as division, each haploid cell divide into two, producing four haploid cell. So meiosis second is similar to mitosis. That's meiosis second is equational division. Here we can see this parent cell which produce larger size, and the replication of DNA takes place. Here formation of two daughter cell which undergoes reduction division due to the reduction division. The formation of two daughter cells, and then this two daughter cell again divide into two of each, and their formation of four haploid cell. So this is outline of meiosis. Then this meiosis, before going to undergo that particular stages of meiosis, it undergoes interphase. The preparatory stage of meiosis and mitosis is named as interphase and is similar in both. It divide into three phases: G1 phase, then G2 phase, and S phase. G1, S phase, and G2 phase. That's similar to the interphase of mitosis. Then meiosis first, and first now we'll see meiosis first. That meiosis first or first meiotic division. It is actual reduction division in which the homologous chromosomes separate without dividing to form two haploid nuclei. It takes place in different steps. That first step is prophase first, then metaphase first, anaphase first, and telophase first. Then this prophase first again having different stages, that leptotin, zygotin, pachytin, diplotin, and dikinesis. So now we'll see prophase first, prophase first of meiosis first. So prophase first of meiosis first is much prolonged and can be divided further into five stages, that leptotin, zygotin, pachytin, diplotin, and dikinesis. First we'll see leptotin. Leptotin which derive from leptoma. This stage of short duration during which condensation and coiling of chromosome takes place. Chromosome become visible as long slender threads bearing numerous bead-like nucleosomes chromosome. Then the identical or homologous chromosome received from male and female parent become discernible. Beside an animal cell, the two pairs of centriole already formed are set free and begin to move away from each other. Each one of them give out radiating fibers called aster rays. A pair of centriole along with the aster rays form aster, as in mitosis. Then the chromosomes are arranged in linear fashion along their length. The nuclear envelope and the nucleolus are prominently visible, the thin chromosome are scattered in the nucleus during this liptotin of prophase first. Then another phase of prophase first, that is zygote. It is also called as zygonema. That the meaning of leptos, slender, and tenin, tenia means band. And then that chromosome become slender and leptotin. Then zygotin. Zygon means tied or pair. Here pairing of chromosome takes place. Zygotin or zygonema, pairing of homologous chromosome known as synapsis takes place during the zygote. Then pairing or synapsis takes place in zipper-like manner and may start at centromere, chromosomal ends, or at any other position. Thus, pairing is highly specific and exact during this zygote. The paired chromosomes are called bivalent or tetrad chromosome. The chromosomes at this stage are shorter, thicker, and more distinct during this zygote. These are the events which takes place during zygote of prophase first. Then next is patchitin. That is also called as pachinema. Patches means what? Thick. That thickening of chromosome takes place during this patchitin. Within tetrad consisting of homologous pair of chromosome, the paternal and maternal chromatids are non-sister to another. Then one sister chromatid takes place and take part and crossing over non-sister chromatid randomly exchange segment or region of homology. Then exchange of information between non-sister chromatid result in recombination of genes. Here in Pachitin, there thickening of chromosome takes place, and here that crossing over takes place, and that exchange of recombination of genes takes place and this patchitin stage. Then next is and this during this patchitin, their crossing over also takes place. Crossing over consists of mutual exchange of equal quantity of chromosomal material between two non-sister chromatid. It involves the different events. The non-sister chromatid taking part in crossing over break simultaneously at the identical point, that is at homologous point. The broken segment are of equal length. The broken segment may rejoin, or there may be an exchange of segment between non-sister chromatids. So here exchange of gene takes place during this patchitin due to the crossing over. This crossing over, during crossing over, the genes located on segment are exchanged between two chromatids. It result in recombination of gene takes place. That recombination of characters takes place. Crossing over does not takes place between sister chromatid. That crossing over takes place only non-sister chromatid. The point where the non-sister chromatids exchange known as chiasma, or it is also called as chiasmata. Then here we can see crossing over takes place during patchiting. These are the non-sister chromatids. Then here we can see crossing over takes place. A.B.Sc chromosome cross with that chromatids of this particular chromosome cross with capital A.B.Sc chromatid, and then there here chiasmata takes place, where crossing over takes place, where joining of that non-sister chromatids. Then after this, there separation of this both chromosome, or this chiasmata gets split, or a terminalization takes place, and here separation of two chromosome. Here we can see exchange of chromosome, that arm of one small ABC chromosome get attached on arm of that green color ABC, capital ABC chromosome. So their genes sequence like capital A, B, and small c. So this is recombination and crossing over takes place during patchitin stage. Then significance of crossing over. This crossing over as most significant. The gametes produced through meiosis receive a new combination of characters or gene. Therefore, individuals with new combination of characters are produced in each generation. Then this form genetic basis for variation and plays important role in evolution. So crossing over is most important in variation and evolution. It is also most important in heredity. Then Next is meiosis. First, that propage first, that is diplotin. Next stage of propage first as diplotin. That is also called a diplonema. The diplotin, diplos means what, double. That doubling of chromosome, or that chromosome become double. So two major events involved in this stage, that is diplotin, that repulsion of homologous chromosome. Homologous chromosome of pair start repelling each other. Homologous chromosome which comes together during zygote, they get repelled from each other, and the homologous chromosome begin to separate and uncoil. The non-sister chromatids which are involved in the crossing over are still held together at the point of crossing over. Then terminalization takes place during this stage of diplotin. The separation and uncoiling of the homologous begins at the centromere and proceed toward the ends. This causes progressive shifting of the chiasmata toward the ends of the chromatids. This is called as terminalization of chiasma. It is also called as unzipping. That during Pachitin stage, many chiasmata takes place due to the crossing over, and this chiasmata get open, and this open like unzipping, uh, and there this chiasmata comes to the end of chromatids, and finally chromosome gets separate. This is called as terminalization. Then next is dikinesis, that another phase of propase first, that is dikinesis. This is last phase of propase first. Chromosome are still in pairs and in contact with each other by terminal chiasma. The chromosome becomes shorter, thicker and more prominent. The nucleolus and nuclear envelope disappear completely. The pair of chromosomes are seen distributed in nucleoplasm. This is dikinesis. Here we can see that disappearance of nucleolus and nuclear envelope, and the pair of chromosomes are seen distributed in the nucleoplasm. In this way, this is what prophase first. Here we can see diagram of prophase first, the interphase, then leptotin, that thin chromosome become slender and having it become the bands, that having bands. Then zygotin, so that pairing of chromosome takes place. Then patchitin, here crossing over takes place, and diplotin, the terminalization takes place, and then dikinesis, the chromosome that disappearance of nucleolus and nuclear membrane, and chromosome become near to each other. This is what diagram which showing different stages of prophase first. The next is meiosis first, that metaphase first. The spindle apparatus of that spindle apparatus showing two types of spindle fibers is formed, the continuous fibers extending from to pole, and chromosomal fiber extending from to the equator of the spindle body. The homologous chromosomes still in pair are arranged at the equator of spindle, and maternal homologous of all pair shows toward one pole, while paternal homologous face the opposite pole. The chromosomal fibers connect the chromosome centromeres of all homologous to the pole on their respective sides. Then anaphase first. Anaphase first, in this phase, the two homologous chromosomes or mates of the bivalents of same chromosome start moving toward opposite pole along the tactile fiber. This process is called as disjunction. Thus, the real reduction is the number of chromosome takes place at this stage. Each pole receiving half the number of original chromosome due to crossing over and prophase. The sets of chromosome that moves to opposite pole consist of recombination of maternal and paternal chromosome. Then here we can see this event of anaphase first and this diagram. Then next is meiosis uh, first, telophase first. Then another phase of meiosis first, that is telophase first. In this phase, the chromosome begin long thread like, that chromosome become long thread like and distinct. The spindle and aster rays gradually disappear and formation of centrosome takes place as in mitotic telophase. The nuclear membrane and the nucleolus reappear, thus forming two nuclei. This is what telophase. Here we can see formation of two nuclei during this telophase first. Then next is cytokinesis. After this telophase first, the cytokinesis takes place. The cytoplasm constrict in the middle or cell plate is formed to form two daughter cells, each containing single nucleus with half the number of original number of chromosomes. Here it may takes place, it may not takes place after meiosis. Interface between meiosis first and meiosis second, if any, is brief. Then generally it is absent. Then in this there is no replication of DNA takes place after meiosis first. Then we'll see meiosis second, that the interkinesis takes place before meiosis second. The time interval between meiosis first and meiosis second is called as interkinesis. The stages of meiosis second are similar to mitosis. The two nuclei form after meiosis first divide during meiosis second and produce four haploid nuclei. So this now we'll see meiosis second. That meiosis second having different stages: prophase second, metaphase second, anaphase second, telophase second. Then in prophase second, in this prophase second, the chromosome again become discernible, each consisting of two sister chromatids still attached at centromere. The nuclear membrane and nucleolus disappear, spindle fiber begin to appear. The aster formation takes place during this prophase second. Then metaphase second. And metaphase second, spindle is completely formed. All the chromosome get arranged in the equatorial plane with centromere attached to the tactile spindle fibers. Then anaphase second, in this the centromere of each chromosome now divides for the first time. The sister chromatids, now called daughter chromosome, travel to the opposite pole. Then telophase second takes place. Then here we can see metaphase second and anaphase second. Then telophase second. The set of daughter chromosome at each pole elongates, become indistinct. The nuclear membrane nucleolus reappear, organizing into nucleus. The cytokinesis further uh, divides each cell into two, thus forming four cells, each containing half the number of chromosomes. So this is a diagram is showing telophase second, production of four nuclei, that products of meiosis, four haploid nuclei. So after this cytokinesis takes place. This is the division of cell cytoplasm. It follows the nuclear division and may be successive or simultaneous. So uh, successive or simultaneous cytokinesis takes place. Then this, what is successive cytokinesis? If cytokinesis takes place after each nuclear division, it is called as successive cytokinesis. If the cytokinesis takes place after both nuclear division are completed, it is called as simultaneous cytokinesis. In this way, we have seen the different stages of meiosis, that is meiosis first and meiosis second, and we have seen different event which takes place during meiosis. And finally the formation of four haploid nuclei after completion of meiosis. Then now we'll see significance of meiosis. Meiosis facilitates a stable sexual reproduction. Due to the meiosis, recombination and independent assortment of homologous chromosome allow for a greater diversity of genotype in the offspring. Then uh, this it is responsible to give rise a large number of variation in offspring due to new recombination produced by crossing over and random distribution of maternal and paternal chromosome. The variation coupled with heredity from a raw material for organic evolution, but for meiosis the evolution of species would be suspended. Then it compensates for doubling of chromosome at genetic fusion by reducing it again to half, and thus help to keep the number of chromosome constant and characteristic of each species. Chromosomal and gene mutation may take place during meiosis. Some of them may be advantageous to the organism and are selected by natural selection. It proves the interrelation between the living organism, since its details are basically similar in all sexual reproducing organisms. Then we'll see comparison between mitosis and meiosis. That mitosis occur in somatic cells, meiosis occur in reproductive cell. That mitosis consists of only one nuclear division, meiosis consists of two nuclear division, that meiosis first and meiosis second. Cytokinesis takes place only once, and mitosis cytokinesis takes place twice, and meiosis, that is simultaneous type and successive type cytokinesis. Two daughter cells are formed during after mitosis, and four daughter cells are formed after meiosis. Dividing cells can be haploid or diploid in mitosis, and dividing cells are uh, diploid in mitosis. Dividing cells are diploid in meiosis. Then in mitosis, there is not involved either pairing of homologous chromosome or crossing over. In meiosis, pairing of homologous chromosome and crossing over takes place during prophase first. Then in mitosis is an equational division. Meiosis is a reduction division. Original character of the chromosomes are maintained in the daughter cell. In mitosis, then chromosomal characters are altered due to the crossing over, causing recombination of genes during meiosis. Then mitosis in mitosis, daughter cells are similar to each other, also to their original mother cell. In meiosis, daughter cell differ from each other as well as from original mother cell. Then mitosis helps in growth and body repairs, and meiosis help in sexual reproduction and regulation of chromosome number in the life cycle of sexually reproducing organism. And this way we have seen that events which takes place during meiosis, that different stages of meiosis, meiosis first and meiosis second, and we have also seen significance of meiosis and comparison between mitosis and meiosis. So we'll see summary of this lecture. Meiosis is a reductional division which helps in sexual reproduction during a first part of meiosis. The chromosome number is reduced to half. The second part of meiosis is similar to mitosis, during which haploid cell duplicate. The end result of meiosis is four haploid cell. Meiosis lead to the genetic diversity, and in this way we have seen this particular chapter, that is cell division. Thank you.